All right. Hello, everybody. Welcome to part 3 of Unit 2. So Unit 2, again, we're talking about the biological basis of behavior. Last in part 2, we talked about neurons and neural impulses and how they kind of fire. Today, what we're going to talk about is neuron communication. Okay. Now, this is really about how one neuron talks to the next neuron. Okay. So we're going to kind of add that piece in a little bit here. Sorry. Now, the first thing I talked about is the synapse. Now, we kind of mentioned this a little bit briefly in some of the other videos, but basically what the synapse is where two neurons meet. Okay, it actually looks like this. This is a synapse right here. So when we have a synapse, there's basically this tiny gap, and this is like a microscopic gap between the axon terminal of one neuron, right? So here's where those axon terminals are, and basically usually the dendrites of another neuron. Right, so here's our dendrite, right? There's these little receptors on this end. This is our axon terminal, which has basically neurotransmitters inside of it. And what's going to happen here is that neurons are going to use neurotransmitters, which are being shown as these little kind of objects, here's little orbs. And those neurotransmitters are going to jump the gap from one neuron to the other, and they're going to basically be sucked into that dendrite, right? Now, this is really, really important, again, because this is how, again, that piece of information gets from one neuron to the next one. So it's electricity all the way until it gets to the end, and then it's a chemical message, right? So that chemical message then jumps this little gap. This neuron, if it reaches threshold, would send that electrical impulse again, that action potential, right? And we'll kind of see how that continues there. So again, our neurotransmitters are jumping that gap of our synapse. The synapse is this whole thing, right? Specifically referring to this small gap in between those two pieces there. So this is kind of a more detailed diagram here, right? So we've got our nerve impulse coming in from the bottom right, that electrical information. And there's some stuff in here that's labeled that we don't necessarily really care too much about, like our presynaptic axon. You know, you're not really have to label that, but it's basically our axon right before it gets to the terminal, right before it gets to the synapse, thus presynaptic. We have what are called synaptic vesicles, right, which are kind of like these little, like, I don't want to call them like air bubbles, but like kind of little bubbles that contain neurotransmitters. We have our synaptic cleft, which is the actual gap, right, between the two. We have, this is a vesicle right here, releasing that neurotransmitter. So these vesicles release these neurotransmitters. You can see the little neurotransmitters. I'm trying to move my mouse so that that thing goes away. Right, you can see the neurotransmitters jumping that gap, right, and moving towards that dendrite, and that dendrite will have little receptors that'll pick it up on the other end. Okay, and we'll see some moving versions of this as well as we go. So, if you are doing one of those coloring pages, now is a good time to kind of stop and just label that synapse, right? So I went through, you can label it, color that in, and then we'll keep going. So, sorry, it keeps moving there. There we go. All right. So let's talk about neurotransmitters a little bit, right? So what are neurotransmitters? So basically what neurotransmitters are chemical messengers that jump this synaptic gap, okay? So they're going to jump the gap between those neurons. So here again is our synapse, right? Synaptic gap or synaptic cleft is right here. Here's our axon terminal, right? This is a dendrite. We've got receptors here. We've got vesicles there. You can see them, right? So all these little guys in the middle, these little molecules floating around, are these neurotransmitters, right? They're chemicals. Now, what they're going to do is basically they're going to, be released. They're going to jump this little gap, and they're going to then bind with a receptor on the other end. Okay? Now, what's really important is that these receptors only fit certain neurotransmitters, right? They bind like a key in a lock, right? So you can't just use your key on any door. It doesn't work. Right? You can only use certain keys on certain doors. All right? And that's really important. So there's basically a whole bunch of different receptors that receive and bind with different neurotransmitters. Okay? Now, while binded, right, the neurotransmitter allows the ions, allows in ions that either excite or inhibit the neuron. Right? So before we talked about how we can have excitatory and inhibitory kind of reactions or messages being sent. Right? This is how that's happening. So some of these neurotransmitters are going to send messages that will excite this next neuron. Some of them will inhibit it. Okay? So there's kind of multiple things that can be happening here. Now, after those transmitters are bind, right? So they jump this gap and they bind with that neuron, they excite it or inhibit it, right? What's going to happen next is those neurotransmitters will be released. Okay? So they're actually going to bind and then they get released. Now there's a few different things that can happen, right? So once those have bound and they've been released, right, some of them will just like drift away, right? As you can tell, this is kind of like a liquidy little area, so they'll just kind of drift away. Some of them will be broken down. Some of them will also go through what's called reabsorption, right? They'll be reabsorbed into that original neuron, right? If they're reabsorbed, that's what's called reuptake. And reuptake is a term that you do want to know. You want to definitely have that definition there, right? So basically, again, it's where a neuron reabsorbs the neurotransmitters that it already released. So it releases those transmitters, they float across, they bind, they excite or inhibit, right? They get released, and some of them will be, reuptaken into that neuron. Again, some of them you can kind of see them there in the background, right? They're kind of like floating away. Some of them will get broken down by like glial cells, right? That's kind of what can happen to all these different neurotransmitters. So just to kind of go through step by step what's happening here, right? So the first thing, and this is our entire neuron communicating, right? So the first thing is that action potential reaches the neuron, right? So the action potential is shot down, it reaches, and it really should say probably the axon terminal, sorry. The action potential reaches that axon terminal so that electricity, right, gets sent down here. What that's going to cause is those neurotransmitters to be released. So the neurotransmitters get released, right? Right here you can see them being released. Those neurotransmitters will then cross that synaptic gap, right? Here they are. And then will bind with a receptor that fits, right? So this receptor, right, is being bound with. Once that receptor has bound with the neurotransmitter, it will either excite or inhibit it, right? So that receptor will either be excited or inhibited. That information then is basically released, right? That excitatory or inhibitory impulse is released. Then the neurotransmitter is released, and one of three things can happen, right? It can be broken down. You kind of see that here, right, where it's being broken down. It can just kind of float away into nowhere, or, right, it can be reuptaken into that original neuron. And there's some other stuff that can happen inside the neuron, but we're not going to really worry about that. So again, those are those steps. So now if we put this all together, right, you can think of, and it's kind of it's a cycle, right? So a neuron is excited, right? It reaches threshold, it shoots the action potential reaches the axon terminal, the axon terminal releases those neurotransmitters, they jump the gap, they bind with that next neuron, they are with the receptor, they excite that next neuron, right? They're released, they go back in, that next neuron is now excited, it reaches threshold, it fires, and it starts all over again. Now, I mentioned before, there's kind of different things that can happen and different things that different neurotransmitters do, right? So we're going to talk about neurotransmitters in kind of general, right, and the kind of the basics of them here. So neurotransmitters can be a little complicated here because they don't always act alone, right? They don't necessarily do what they do entirely on their own. And they actually, a lot of times, a neuron might be receiving a bunch of different neurotransmitters at once, right? So they can be receiving inhibitory and excitatory messages at the same time from a bunch of different neurons all at once, okay? Now, we do know that, and then they also all will function with each other, right? So it's not like we necessarily release only one neurotransmitter at a time. Even one neuron can be releasing multiple kinds of neurotransmitters, right? That'll kind of act together and then have different effects depending on how much of this and how much of that is kind of being released. Now, we do see that some neurotransmitters are more or less common in certain areas, right? So something like serotonin, which is connected very much with like the feelings of pleasure. Serotonin, right, is a lot of times will be very high concentrations of it will be found in things like what's called like the pleasure pathway, right, which is in our brain, and it's basically like the part of our brain that's really connected with what feels good, right? Now we might have something different like maybe like adrenaline, right? And adrenaline, obviously, right, is going to be maybe more focused in our muscles, right, because that's to do with movement. So we're going to see differences, but they are kind of spread out. But we might see, you know, more here or less there, depending on what kind of thing it is, right? And we'll talk about what the specific neurotransmitters are in class specifically. So we can also break them into two groups, right? Excitatory or inhibitory, right? So excitatory is, again, exciting the next neuron to fire, right? So you can think of them, right? Right here, they're just yelling to fire, right? Fire, fire, fire. Our inhibitory neuron, right, is telling us not to, right? It's saying no, don't fire. It's trying to stop that next neuron from firing. Okay. So again, we have these two sides, right, that we can kind of see. Now, there's also a really important connection between neurons and drugs, right? So the reason that drugs do anything to us at all is because of the way that our neurons function, right? And specifically what they do with neurotransmitters, right? So generally speaking, we've basically got kind of like three types of drugs, right? Now, again, it's really important to understand that when we say drugs, it literally means anything that affects our body, right? So you can think of, obviously, like illicit drugs. You can think of things like alcohol. You can think of medication you get from a doctor, right? Basically anything, right, that has an effect on our body is technically, right, could fall in this kind of category here. Okay, so there's kind of like three groups. So we've got agonists and antagonists, right? So agonists increase the neurotransmitter action by increasing production or release, by increasing the production or release or by blocking reuptake. So basically, drugs that are agonists, right, are going to cause a neurotransmitter to either be like released a lot more for our body to make more of it, or to stop it from being pulled back into the cell. And that's going to cause that neurotransmitter to basically be overreacting, right? To be having a bigger impact. So an example, and we'll watch this video in class because we can talk about it there, and I'll post the video directly in there. One example they show us, I believe, is like cocaine, right? So one of the things that cocaine does is it pushes out, right? It causes the release of things like serotonin and dopamine, right? And so what happens is all that neurotransmitter gets dumped in, and that's why we get the effects we do from all these different drugs that we have, right? So for this one, it's that idea of feeling like super euphoric, right? Comes from that release of that chemical. We see the same thing with something like ecstasy, right? Where it's causing this, I believe ecstasy also blocks reuptake, but it's causing this basically dump of dopamine, right? And that dopamine makes us feel good, so then we feel really, really good, right? And that's kind of what's happening with these agonists, right? So again, agonists are increasing how much a neurotransmitter is doing. Now there's also antagonists. Now antagonists are going to do the opposite, right? It kind of makes sense. Agonists, antagonists. Antagonists are going to decrease neurotransmitter action. Now the way they're going to do this is generally by either blocking the receptor or blocking the release of that neuron, right? So it'll block these kind of vesicles and we won't be able to release that neurotransmitter, or it'll block the receptors and then we can't, receive that neurotransmitter, right? So we've got, again, our agonist or antagonist. I believe, and I might be off on this, but I believe like alcohol is an antagonist, right? And it's blocking those neurotransmitters from firing, which is why we have, again, some of those effects, right? It's things like slowed speech and lack of coordination, right? Is because I believe it's because it's blocking some of these receptors or blocking the release of some of those neurotransmitters that have to do with our movement, right? And our ability to move around effectively. We also have some drugs that actually mimic neurotransmitters, right? So they're close enough chemically where they can bind with the same receptor that a neurotransmitter can, right? So I believe, and there's an example of this in this video that we'll talk about as well. I believe the example, I believe it's meth actually. So meth is close enough to actual neurotransmitters we have in our body that it just actually will bind with the receptor itself, Now this can cause a whole lot of other problems, right? Because if it's binding, right, that means that we're obviously experiencing whatever that neurotransmitter is going to be. Sometimes, depending on what the drug is, it can actually like break. I don't know if break's the right word, but like overstimulate or break the receptors, which will cause problems later on because you won't be able to receive those neurotransmitters anymore. Sometimes it, causes stuff to get stuck, right? So again, it's just kind of 3 different ways that these are kind of impacting, right, what's going on. And again, all of them, the only reason these things work on us and any medication works on us is because of how these neurotransmitters function, right? So whenever you take any medication, basically what's happening is chemicals are being put in your body that are then impacting in some way those neurotransmitters, right? Through agonists, antagonists, or, right, they could be mimicking as well. Okay, so if you would, please do a quick summary for this conversation about neurotransmitters, right? Again, we will specifically talk about each neurotransmitter because you'll need to know what they are, but we'll do that in class, and just kind of do a short summary for me.